Hello everyone, the third lab of practical microbiology for the second stage anesthesia department will be about catalase test and coagulase test. Catalase test is a simple test to determine the production of catalase enzyme by some species of bacteria. So this enzyme produced by many other organisms, but we have microbiology and some bacteria produce catalase enzyme. This enzyme protects the bacteria from hydrogen peroxide, which is hazardous, and it's H2O2 that can damage and kill the bacteria. But if the bacteria produce this enzyme, it can protect itself from this substance, which is hydrogen peroxide. We have most aerobic bacteria and facultatively anaerobic bacteria can produce catalase enzyme, while obligate anaerobic bacteria, they cannot produce this enzyme. The aim of catalase test, it's a way for characterizing bacterial species, and it can be used to classify and identify the bacterial species. For example, we have two main groups of bacteria, which are Staphylococci and Streptococci. Both of them are cocci bacteria and gram-positive cocci bacteria. But if you want to differentiate between these two groups of bacteria or these two genus of bacteria, because each of them have many species, we can use catalase enzyme to differentiate between them. Staphylococci bacteria are catalase positive, while Streptococci bacteria are catalase negative. The catalase test also used to differentiate between the gram-positive bacteria bacilli. For example, we have Clostridium bacterium. The aerotolerant strains of Clostridium, they cannot produce catalase enzyme, which are catalase negative, while the bacilli, they produce catalase enzyme and they are catalase positive. Both of them are gram-positive bacteria, bacilli, and spore-forming. The principle of this test, the principle is firstly start by adding a drop of H2O2 to bacterial placed on a slide. Then, if the bacteria produce catalase enzyme, the H2O2 will be broken down and it will change to water and oxygen gas, resulting in the production of bubble. If we have production of bubble, or if we have bubbles after mixing these two components, the bacteria with H2O2, it means that it is catalase positive. But if we don't have bubbles, it means that that bacteria cannot produce catalase enzyme. And this is the equation of this reaction. We have two molecules of H2O2 in the presence of catalase enzyme will change to two molecules of water and one molecule of oxygen gas. Catalase test method, we have two methods. We have slide method to apply this test, and also we have tube method. And here the result of slide method, this is catalase positive and this is catalase negative, while the result of tube method, we have catalase negative like this in the tube, while catalase positive will be like this, and here's are bubbles of that indicate it is positive for catalase test. The procedure of this test, we will obtain a clean microscopic dry glass, slides, and a bottle of hydrogen peroxide. We will place a drop of H2O2 on the dry slide. By using sterilized wood stick, we will add a small amount of the bacteria to the H2O2, then we will mix it very well. Then we look for the bubble formation. If we have bubbles, immediately it's good. If we don't have, we will wait for 5 to 10 seconds. We will see if there's bubble, it means it is positive. So the result, if we have bubbles, it means catalase positive, and that means that we have Staphylococci bacteria. Well, if there is the bubble absent or there was not bubbles, it means catalase negative, such as we have Streptococci bacteria. Here we can see if we add a drop of normal saline of water and mix it with the bacteria, bacterial colony. Then we will add the same amount of 3% H2O2, which is hydrogen peroxide, and we will mix it. If there is bubble, it means it is positive like this. If we don't see the bubbles, so it means it is negative, and maybe that bacteria be streptococci bacteria. We have some precautions of catalase test. First of all, the tested organism should not be taken from the blood agar culture. Why? Because we have red blood cell on blood agar. Naturally, they produce or they have catalase enzyme so that they will give us false positive results. If that bacteria do not produce catalase enzyme, will be positive because they are cultured on the blood culture and the blood naturally have catalase enzyme. Also, we have H2O2, should be fresh as it is very unstable and can react easily with any other molecule or substance in the environment, and it should be covered immediately. We have also, we must prevent iron wire lock using, and those are the precaution of catalase test. We have another biochemical test, which is coagulase. It's also an enzyme. So coagulase test is a type of biochemical test. It's used for differentiation between the Staphylococcus aureus, which is coagulase positive, and other Staphylococci species such as Staph epidermidis and Staph saprophyticus, which are coagulase negative. We have many other species of the Staphylococcus bacteria, but those are the main medical important species. The other Staphylococci such as saprophyticus and epidermidis or staph epidermis, they do not produce coagulase enzyme. So coagulase, as we said previously, is an enzyme, protein enzyme. Their composition is consists of protein, which produced by many microorganisms, which help in the conversion of protein in our body or in our blood plasma, which is fibrinogen. It helps them to convert the fibrinogen to fibrin. Those are proteins that related to the clotting or clotting of the blood. The coagulase containing microorganisms usually contain a protective barrier around themselves that increase their pathogenicity and resistance against the immune system. When that bacteria produce coagulase, so they change the fibrinogen to fibrin. When they change the fibrinogen to fibrin, they make as a protective barrier around themselves, and this will increase the pathogenicity and resistance from our immune system or against our immune system. This enzyme, as we said previously, is secreted or produced by Staphylococcus aureus, which this will contribute to the virulence or increase their virulence factors and involved in forming a fibrin wall that surrounds the Staphylococci lesion. Type of coagulase enzyme. Staphylococcus produce two forms of coagulase enzyme, which is the free form and bounded form. The coagulase free form or the free coagulase enzyme is a heat labile, which is secreted extracellularly. It's free, so it secreted from the bacteria to the extracellular of the bacteria, and it can be detected by tube coagulase test. So we have two methods of coagulase enzyme, tube coagulase test, and we have also slide coagulase test. While bounded coagulase is a heat-stable enzyme that cannot change their structure easily. It's attached to the bacterial cell wall and react directly with fibrinogen and convert them to fibrin, causing the bacterial cell to clump with plasma. So it's detected by slide coagulase test, while free coagulase type is detected by tube coagulase test. The principle of coagulase test, the enzymatic protein coagulase, as we said previously, is enzymatic and it consists of protein, is thrombin-like substance. Their structure is chemically like thrombin. Thrombin also is a protein found in the plasma of the blood. It helps to convert fibrinogen into fibrin, which results in the clotting or clumping. So Staph aureus carry two different types of coagulase, which may be in the free form or bounded form. The procedure of coagulase test, the slide test, we will apply it in the lab to detect the bounded or bound coagulase. So that coagulase that are attached to the surface of the bacteria and react directly with the plasma to make fibrin. We will put a drop of normal saline on the slide, then we will mix it with 1 or 2 colony of the bacteria that will be tested. Then we will add an equal amount of the plasma. The plasma will obtain it after the blood taking from a patient and centrifuging in the trisodium citrate tube. So we will obtain plasma, and that plasma we will use it in this test, and we will mix it with bacteria. The positive result appear as clump within 5 to 10 seconds. The bounded coagulase is also known as a clumping factor, and this is the shape of the result after we apply the procedure. This is Staph epidermidis, which is negative for coagulase test, while Staph aureus, those both sample are for the Staph aureus. We have clumping here, and those indicate that the coagulase test is positive. We have also the procedure of the tube method to detect the free coagulase. We will make dilution of the plasma in 1 to 10 normal saline. So we will add or mix 0.2 ml of the plasma with 1.8 ml of the saline or normal saline. We will pipette from that dilution 0.5 ml into a clean tube. After that, we will add 5 drops, 0.1 ml, about 0.1 ml of the tested organism to the tube after labeling it. So we need broth here, and we will add from that broth culture 5 drops to the 0.5 ml of diluted plasma in the tube. After mixing, we will incubate for 30 seconds at 35 to 37 degrees Celsius, and we will examine after 30 minutes if there is no clotting. So we will examine at interval for up to 6 hours, and the positive result lead to gel formation like this. Here we can see there is gel formation at the bottom of the tube, while here the result is negative. There is no gel formation or clumping. Catalase test. For catalysis we need bacterial culture, we need hydrogen peroxide, and disposable wooden stick, and also we need dropper and clean slide. This test used for distinguishing the Staphylococcus species from Streptococcus species. The hydrogen peroxidase must be in dark bottle and covered. Now we will start the procedure. We will add a drop of hydrogen peroxide on a clean slide. Then by clean wooden stick, we will add a colony of bacteria. Then we will mix it with hydrogen peroxide. The result, if it's positive, we have these bubbles as you see. If it's negative, the bubble will not appear, and this is positive. It means it's Staphylococcus species. Today we have coagulase test. Coagulase test is a test or an enzyme produced by some species of Staphylococcus, especially we have Staphylococcus aureus, and used to distinguish Staphylococcus aureus from other species such as Staph epidermidis and Saprophyticus. In this test we need bacterial culture, we need normal saline, we need plasma, and clean slide and droppers and wooden stick. Firstly, we will add a drop of normal saline on a clean slide. Then we will add a colony of a bacteria by clean wooden stick, and we will mix it with the normal saline. After that, we will add the same volume of plasma and we will mix it by wooden stick. If there is clumping, it means positive, and if there is no clumping, it means negative. We will wait for a few seconds, and now we have clumping. They appear here, so it means it's Staphylococcus aureus. Here, this is clump. This is the particle of the clump.